We are Group 10, and we will be presenting gel electrophoresis.
Gel electrophoresis is a method for the separation of DNA, RNA, and proteins, as well as a way to analyze these samples based on size. When voltage is applied to the gel loaded with a sample, the molecules will migrate to the electrode of opposite charge. Shorter, smaller molecules move faster and further through the gel compared to larger molecules. In this experiment, we will be observing the migration of protein samples where the charge is dependent on the pH where the protein is found. The objective of this experiment is to identify 3 unknown protein samples using a standard sample with known molecular weights and analyze the accuracy of these findings.
There are 2 layers to the gel. First, there's a resolving gel and then a stacking gel. The resolving gel is the main large area of the gel, and the stacking gel simply sits on top of it, and it is where the comb is placed. The resolving gel has a higher concentration of acrylamide, making the protein separate more as it is harder to move through this more dense gel. The stacking gel promotes the initial movement of the proteins to get things started. In addition, since the buffer is basic with a pH of 8.3 and the stacking gel is more acidic with a pH of 6.3, this allows the proteins to become neutralized before they start to move through the gel. The resolving gel is added to the mold until it reaches a dot marked about 1 centimeter from the top. Propanol is then added to remove any bubbles within the solution.
Once the resolving solution has polymerized and has been rinsed with isopropyl alcohol, the stacking solution is used to fill up the remaining space up to the top of the small plate. The comb can then be inserted and the gel allowed to fully polymerize. It's okay if some of the solution spills out when inserting the comb. Allow the gel to polymerize for 45 minutes.
The protein samples are prepared by adding SDS buffer and beta-mercaptoethanol to the vials. This decoils and disrupts the protein structure, partially denaturing the sample. Then, the samples are heated in a dry bath at 95 degrees Celsius for 5 minutes to completely denature them and break up the disulfide bonds. When placing the gels into the chamber, ensure that the short glass plates are facing inward, otherwise the current will not flow. After the gels and buffer have been loaded into the chamber, carefully load the proteins into the wells. To place the protein samples into the gel, you carefully place 15 microliters of the proteins into the wells. Make sure the tip of the pipette is between the glass plates and is inside the well before injecting the protein. Once the protein samples have been finished being loaded, start the apparatus.
Once the migration front has reached the bottom of the gel, the device can be shut off and the gel removed from the chamber. The Biorad imager tray should then be lightly coated with buffer and the gel gently placed flat on the middle of the tray. The tray is then inserted into the imaging machine and the software is run. Using the Biorad Image Lab software, the protein bands can be analyzed and their molecular weights can be automatically estimated. The Volume Tools tool can be used to manually draw boxes around each band. This manual method produces the most accurate results for molecular weight estimation. The software is then able to determine the intensity of each band and the relative quantities of protein within them, as well as produce a table listing all of this information. Using this information, you can calculate the molecular weight of the protein using a weighted average of the bands in each lane. Once you obtain the molecular weight, you can compare the value to similarly sized proteins in a database and search for the one that most closely matches your protein and is related to the type of sample from which the protein bands were separated from.
In summary, in this experiment, we used gel electrophoresis to make our predictions about the identity and quantity of our unknown proteins. Using the standard as a calibration, the imaging software was able to determine the molecular weights of each band, and using that information, we were able to identify the molecular weights of our proteins. For protein A, we identified that it was about 29.55 kilodaltons, and it was a chloroplastic. For protein B, we identified that it was 13.5 kilodaltons, and it was aurictologous. And for protein C, it was 128.8 kilodaltons, and we identified it as bovine serum. And for the known Z at 30.2 kilodaltons, which is similar to A, we also identified that as a chloroplast. So we found that Z and A were the same protein according to our results. Although we have made these conclusions on which protein corresponds to our unknown, in reality it is unrealistic to predict the protein based solely on the molecular weight, and we would need more information and more things to corroborate this data with.